Our final major topic today is excretion. We'll start with an introduction and then we'll get into the specifics of mammalian and in particular human excretion. The idea here is that we produce a lot of waste products via all the processes in our body. So we have not just cellular respiration, but we have the breakdown of waste products like proteins and other materials, cellular components that wear out over time. And this leads to a lot of waste. So we know that we make water and CO2 as a byproduct of respiration. We also make material called bilirubin through a lot of different processes in our body. And then there's a class of waste called nitrogenous waste that we get when we break down proteins. We have to get rid of excess nitrogen. All of this stuff is going to be lost either via respiration, like water and CO2 we exhale out via respiration, or via filtration from the blood followed by excretion. And that's what we're going to look at right now in the context of nitrogenous waste. So what on earth is nitrogenous waste? Well, the idea is that we can take proteins and nucleic acids and break them down for energy. They get converted either into fats for use later or carbs for use in the short term. Well, both proteins and nucleic acids contain lots of nitrogen. Think about the structure of an amino acid or a nitrogenous base in DNA or RNA. So we have to deal with the nitrogen in some way. The process of removing those nitrogenous groups is called deamination. It's the process of removing an amino group, an NH2. By the way, this is an aside. Ask me about deamination sometimes. I've mentioned previously that I love cytosine in the context of viral evolution. It's like I really love cytosine. And the reason I really love cytosine is because of a reaction that it does called deamination. So if you're ever curious, ask me about deamination and cytosine, and then be prepared because I could literally talk for hours about that one specific thing. Just that's a random aside. Deamination, I love deamination. It's so interesting. Okay, back on topic. Now the problem here is when we do deamination, we chop off those amino groups and they get converted to NH3, which is ammonia. And what do we know about ammonia? Yeah, it's highly toxic. So we now have to deal with the ammonia that we just generated inside our own body. So this ammonia is the basis for the nitrogenous waste we're talking about. Different organisms are going to deal with it in different ways. And there's really three types of nitrogenous waste that different organisms secrete. The first is just ammonia. So like fish, for example, if you live in the ocean, you could just secrete ammonia into your environment. You don't have to store it in your body at all, so it never accumulates to the point where it's going to be dangerous. It's highly soluble, so you just release it into the surrounding environment. It dissolves in the ocean. It's never going to accumulate to the point where it's dangerous because you're in an ocean or you're in a river or a lake or whatever. It's never going to get to the point where it's dangerous. You just secrete that ammonia directly and it's gone. This is good because it requires very, very low energy. You don't have to do any additional processing. If you're on land, that's not an option. So plan B is to make urea. This is what we do. So we see this in mammals, amphibians, sharks, and some fish actually make urea. Urea is great because it has very low toxicity, so you can store a lot of it. Like we store urea in our urinary bladder, and that's okay because it's not toxic. Whereas if we were to store ammonia in our bladder, that would be really dangerous. The downsides here are, one, it requires energy to make. Obviously, this is a bigger molecule than just ammonia, so it requires energy to make urea, and you need to excrete it using water. So you can't just release the urea on its own. You need to use water to get rid of it, and as we saw, this could be a problem for living on land because you need to conserve water as much as possible. The last option is to use a material called uric acid. Uric acid is what we find in reptiles and birds, insects, and, for whatever reason, land snails. Now, what the reptiles and the birds and the insects have in common is that their eggs have a lot of yolk in them, and they have very complicated development in that egg. And the important thing with uric acid is that it is insoluble. So it's not going to dissolve within the aqueous solution of the egg. It's going to fall out of solution. Now, uric acid is still relatively low toxicity. It's not as safe as urea, but it's relatively low toxicity. But if it was to accumulate in an egg, for example, think about like a chicken egg. If that uric acid was to accumulate within the aqueous solution of that egg, it could potentially poison the embryo. This is where the insolubility comes in. This is really important because it falls out of solution and accumulates in one of those extra embryonic membranes rather than dissolving and potentially poisoning the developing embryo. So this is a really good option if you have a self-contained egg that's going to develop for a long period of time. You know that uric acid is going to fall out of solution and not be dangerous to the embryo. The other side of this is because it's insoluble, you don't need to lose a lot of water getting rid of it. So think about like bird poop. A lot of that is going to be uric acid. There's not a lot of water to that. It's usually pretty dry, and that's advantageous if you're living on land. The downside to all of this, of course, is the high energy requirement because this molecule is much larger than urea. So you need to use even more energy to make uric acid. So those are the three types of nitrogenous waste and kind of the cost-benefit analysis for each one. So we're now going to focus on the process of excretion, specifically in mammals. The process of excretion has 4 steps. The first step is called filtration. This is where you're actually taking stuff out of the blood. Basically, the blood interacts with a layer called transport epithelium. And this is going to be a layer of either one cell layer or more than one. And the point is that it's a one-way street. So the blood encounters that filtration layer, the transport epithelium, and material can move from the blood into the filtrate, but it can't go back the other way. This process is not specific or selective at all. It's basically just based on the size of the molecules. So big molecules like proteins can't get through that transport epithelium, but smaller molecules like ions and water can. So it's more or less based on size here more than anything else. In this very simple figure, the filtration step is happening up here. Following filtration, step 2 is reabsorption. This is where useful substances are moving from the filtrate back into the body fluid, which is usually the blood. In the context of mammals, it's blood that we're talking about here. Okay, so we're moving things that are useful from the filtrate back into the bloodstream. Then we have secretion. Step 3 is secretion, which is active transport of waste products from the blood into the urine. So this is really important to get rid of toxins, to get rid of excess ions, and to get rid of solutes that we just don't need. Secretion must be via active transport. And I want to make that distinction very clear. Reabsorption can be via active transport, or it can be via facilitated diffusion. It can go either way. But secretion must be via active transport because the point is to get rid of stuff rather than reach an equilibrium. If you have too much of a specific ion in your blood, you don't want it to be at equilibrium with the filtrate. You want to get rid of all of that ion. So you use active transport to pump it against the concentration gradient into the filtrate. Okay, so that's your third step, secretion. That's for getting rid of stuff, and it has to be via active transport. And then our fourth step is excretion. So this is getting rid of the filtrate. For mammals, this means getting rid of it in the form of urine. So let's review before we get to the human excretory system. Our first step, which takes place up here, is filtration. Then we have reabsorption, which can be active or passive to take back stuff that we want. We have secretion to get rid of additional bad things. That has to be active. And then to get rid of the filtrate from the body, we have excretion. The last thing we're going to look at today is the human excretory system. Now there is a lot of detail going on here. I don't want you to worry about most of this detail. I'm going to highlight things as we go. We're going to see different parts of this figure over the next few slides. I'm going to tell you what's important, and I want you to not worry about the rest of the stuff. The primary organ involved in human excretion is the kidneys. This is the main mammalian excretory organ. For humans, it's a pair of bean-shaped structures, so they're in kind of your lower back, right below your adrenal glands. Kidneys have 2 primary functions in the body. The first is to regulate the blood composition, so 3 of the 4 steps that we just looked at are going to take place in the kidneys. The second thing that kidneys do is produce the urine that we'll lose during the 4th step, the excretion step. Now, kidneys have 2 main layers to their structures, and while this is new for kidneys, the terminology is actually not new. We've seen these words before. So we have the renal cortex, which is the outer layer of the kidneys, and the renal medulla, which is the inner layer. Beyond those two words, inner and outer layer, you don't need to know more about the cortex versus the medulla. They are very important functionally in terms of like concentration gradients, but don't worry about the specifics. Just know that just like the adrenal glands, the outer part is the cortex, the inner part is the medulla. If we zoom in, so we're going to take like a little pie slice out of this kidney right here. If we zoom in, we can see a structure called a nephron. So that's where filtration and reabsorption and secretion actually occurs. It takes place in the nephron. There are two types of nephrons. I don't care. Just know the term nephron. What we're specifically talking about is referring to what's called a cortical nephron, but I don't care about the distinction between the two types of nephrons for our purposes. Just know what a nephron is and functionally what it's doing. So let's look at what a nephron is and functionally what it's doing. So first, this is the nephron. So this kind of beige tube here, this is the nephron. Surrounding it, we have blood vessels. That's in purple. Those are the blood vessels that are doing exchange with the nephron. But the nephron is the part of the kidneys that's actually making and then moving the filtrate. At the front end, the closed end where the nephron tube starts, we have a structure called Bowman's capsule. So this is Bowman's capsule. You can think about it like a sphere with a little opening on one side. That's Bowman's capsule. That's kind of the dead end where the nephron starts. And inside Bowman's capsule, we have a spherical network of capillaries called the glomerulus. Okay, so you've got the glomerulus or the blood vessels inside Bowman's capsule. That is where filtration occurs. So the first of the 4 steps of excretion is filtration, and that occurs between the blood in the glomerulus and the Bowman's capsule, which is the beginning of that nephron tube. So in this picture, blood is going to enter the glomerulus. Materials in that blood are going to be filtered out across the transport epithelium into the nephron via Bowman's capsule. Below Bowman's capsule, we have a structure called the loop of Henle. So this is, you have this tube, winds back and forth, and then it dips down into the medulla, and then back up towards the cortex. That loop down and then back up is called the loop of Henle. You don't have to worry about like the different names of the parts of it or anything. Just know that the loop of Henle is where the processes of reabsorption and secretion are occurring. So you have exchange between the nephron as it goes down and back up and the surrounding capillaries at the loop of Henle. Relating this back to our simpler diagram before, this is a representation of what's going on in the kidneys. So we have our blood vessels. Up here we have a glomerulus in a Bowman's capsule. And then this region down here where we have reabsorption and secretion, that is going to be the loop of Henle. I apologize if you can hear a dog barking in the background, but I got to get this done. Sorry. So that leaves just one more part of this process, and that is excretion. So the fluid that we're making in the kidneys is urine. This is going to be an aqueous solution containing urea, lots of water, and other dissolved waste products. It's continuously produced by the kidneys, and it's going to move through a series of structures. So it goes from a tube called the ureter from the kidneys into the urinary bladder, where it can be stored. The urinary bladder can contain up to 1.5 pints of urine, and then it will exit the body via the urethra. That is the process of excretion, the 4th and final stage in the broader process of excretion, or the functioning of the excretory system. So let's summarize excretion. The idea here is we need to have structures that are going to regulate the water balance in the body and remove nitrogenous waste. For vertebrates, the structures that do this are the kidneys, and they are very highly adapted to remove solutes and transport those solutes back and forth to get exactly the right mix in your blood while also minimizing water loss.